Hey guys, welcome back. Today we're going to be going over the cell cycle, which is the second lesson of Unit 3. Also, we're celebrating my dad's birthday tonight, so I hope you like my super cute little birthday hat. Hopefully it's not too distracting for you. So like I said, we're going to be going over the cell cycle today. So we're going to be talking about all the different phases that make up the cell cycle. Then we're going to kind of dive into that and look at the different parts of interphase. We're also going to briefly touch on what the difference between mitosis and cytokinesis is, as well as something called G0. So the cell cycle is just all of the different phases of growth and division that a cell goes through in its lifetime. There are two main parts of the cell cycle. There is interphase, which is the growth portion of a cell's life, and it makes up the majority of the cell cycle. Most of a cell's lifetime will be spent in this growth phase. And then there's the division phase, where one cell divides into new cells. And then each one of these phases does have different sections. So in interphase, again, this is where the cell is going to spend most of its life. About 90% of a cell cycle is interphase. All of the normal cell functions are happening here, like cellular respiration, so making ATP, metabolic processes, so like, for instance, breaking down proteins into amino acids. And it's also when the cell prepares for division. During this interphase, the chromosomes are uncondensed, so we actually call them chromatin at this point. And then interphase can be divided up into three different sections. There is G1, also called gap 1 or growth 1. Then there's the S part, which is stands for synthesis. And then the third section is G2, which stands for gap 2 or growth 2. So in G1, that is the start of interphase. The cell is going to be very small because it just came from dividing in half from another cell. And then during this portion of the cell's lifetime, it's going to grow very quickly. And again, all of those normal cell processes like making ATP are also happening. Then we get to the synthesis portion of interphase. And the S part is really important because it's when chromosomes are replicated. So what I mean by that is that it's when chromosomes go from all being single-stranded to all being double-stranded, where they make their sister chromatids. And again, once you have those sister chromatids, they are held together in the middle by a centromere. This needs to happen before cell division can occur because you want to be able to then rip apart your sister chromatids, put them into different cells, and each cells end up with the same chromosomes that you started with. So you started with a single chromosome, then you doubled it, and then you split them apart and they're back to single chromosomes in separate cells. After the chromosomes replicate, we then move into G2 of interphase. This is where the energy reserves of the cell increases. So it's going to, again, make more ATP. It's going to start storing more glucose. The cell is also going to make proteins and any other structures that it's going to need for division. And it's going to look at any mistakes that might have happened during chromosome replication or DNA replication and try and fix those mistakes. And there's also a little bit of growth that happens in this phase as well, though not as much as what happens in G1. So those three parts, again, make up 90% of the cell cycle. That is the interphase. The remaining 10% of the cell's life cycle is spent in this division phase. So this is where the cell is going to divide. In somatic cells, which again are the normal cells of your body, each cell will divide once in order to form two cells. Both of those two new cells will be identical to each other and identical to the original. And that process is called mitosis. We'll talk more about that later. In your sex cells, so your sperm and egg, the cell is actually going to divide twice to form 4 cells. Each of those cells will be different from one another and different from the original. Each of those cells are also going to have half as many chromosomes as the original. So the original cell will be diploid, the 4 cells that are produced are going to be haploid. And this process is called meiosis. So either meiosis or mitosis will happen depending on the type of cell, and then cytokinesis will follow. So with either mitosis or meiosis, you are taking the genetic information and dividing it into different groups. If it is mitosis, the genetic information in the original cell gets replicated, so it gets duplicated, and then 1 copy of the original gets sent to each new cell. So the 2 new cells are identical to each other and the original. And this is done just by, again, you start off with a single-stranded chromosome, you double it, so now you have double-stranded chromosomes, and then you separate the double-stranded chromosomes into the 2 sisters. Now those are 2 separate single-stranded chromosomes, but they are identical to each other, and so when they go to different cells, those 2 cells are then identical to each other. Again, this happens in all of your somatic cells in order to make sure that when cell division happens, which is constantly happening in your body, you're not changing who you are. Your eye color doesn't change, or your skin color doesn't change every time you make new skin cells, for instance. And then meiosis is when you divide the genetic information into non-identical groups, and again, also cut the number of chromosomes in half, so go from diploid to haploid. And that is done in the sex cells to make sure that all of your gametes are unique, so that all of your offspring will be unique. And also make sure that the sperm and egg have half the number of cells of a normal body cell. So when they fertilize each other, you go back to having the normal amount of chromosomes. And we're going to go more into depth into both the process of mitosis and the process of meiosis in a later lesson. Whether you have mitosis or meiosis, though, both of those refer to just the division of the actual genetic material. So once the genetic material separates inside of the cell that's dividing, you then have to divide the rest of the cell. So you have to divide the cytoplasm and all the organelles. So that is what cytokinesis is. It's all of the other parts of the cell dividing to create the actual 2 new separated cells, or 4 in the case of meiosis. So with mitosis, cytokinesis happens at the very end. With meiosis, it'll happen halfway through and then again at the end. So it happens twice as often in meiosis. These are all the reasons why cell division needs to happen. So we already talked about the first one here, growth from a zygote into a multicellular organism. So when you have the sperm and egg first come together, the sperm fertilizes the egg, right away you just have a single-celled organism. And then we have the cleavage process start until we make a morula. And so that process of making the morula, that cleavage process, is cell division. And cell division will keep happening at a fairly rapid rate the entire time that that embryo is growing from the inner cell mass to the embryo to the fetus. The way that that organism keeps getting bigger and bigger and bigger is through the process of cell division. Also, whenever your cells die or are worn out, cell division is used to replace them. So for instance, we replace our skin cells quite frequently because they get worn out and fall off and die all the time. So when you are in need of more skin cells, one of the non-dead skin cells will duplicate so that you have 2 more. This happens with most of the cells in your body, all at different rates. Also, if you have any damaged tissue, so for instance, if you cut yourself and that cut needs to heal, then your skin cells surrounding that cut will divide and replicate until that cut is healed over. And then also to produce sperm and egg. With egg production, you have the follicle cells dividing during that whole follicular phase of the menstrual cycle. Though you don't actually divide to get new eggs as a grown woman. But males are constantly producing new sperm through that process of spermatogenesis in the seminiferous tubules. So cell division is used for that throughout their entire lives. With that said, we do have some specialized cells in our body that never divide. They don't ever go through the cell division phase of the cell cycle. And instead, they do their kind of like growth phase. They'll do part of interphase, and then they'll go into something called G naught, which is how we say this little G with the O beside it. We call that G naught. So yeah. So G naught, it's not actually a phase of the cell cycle. Instead, it's kind of like a little off-ramp of the cell cycle. The cells that enter G naught phase stop growing. They aren't preparing to divide. They're not planning on dividing. Sometimes this is just temporary and the cells will go back into the cell cycle afterwards. But many cells stay in G naught permanently after they finish going through their growth phases, and they won't ever divide. So a lot of neurons are like this. That's why if you sever your spinal cord or something, as of right now, we don't have really great treatments to get like your legs working again, because unlike if you cut your skin, those cells can divide and repair. Your neurons in your spinal cord, for instance, aren't able to divide and repair that damage. Though they are looking at the use of stem cells to repair spinal cord injuries, but that is a different topic. So this is what the cell cycle looks like. So again, it starts in this G1 here, where it starts like growing and stuff like that. After some growth, again, some cells are going to go into that G naught and just spend the rest of their life there, but most cells will continue on and get into S phase, where the chromosomes are duplicated. So as you can see here in the G1 cell, we've got 2 single-stranded chromosomes. And then by the end of S phase, we have 2 double-stranded chromosomes. And then in G2, again, it's just checking to see if any mistakes happened during chromosome replication and fixing any mistakes. And also still a little bit of growth. And then at the end of G2, we get into mitosis or meiosis. So this brown and purple section here are both the division phase. If the division phase includes mitosis, we'll make 2 new identical cells. If it was meiosis, we would just make 4 cells here instead at the end of cytokinesis. Okay, that's it. We're going to be talking more about a lot of this stuff later on.